Good afternoon, everyone. Welcome to today's lecture titled Making Animals Tell Their Stories by Professor Anindita Bhadra. Before we begin, I would like to say a few words regarding the initiative by the Office of Communication, Indian Institute of Science. The lecture is being held under the initiative Paraspar of Office of Communication. Very often, we only know about the product of knowledge. Through Paraspar, we try to unravel the journey of knowledge production. The present lecture series, Journey of Discovery in Science, will discuss the methodologies and processes in science. We are glad that as part of this lecture series, today we have an alumna of our institute who has achieved wide recognition for her work. Dr. Anindita Bhadra. Dr. Anindita Bhadra is the co-chair of Global Young Academy and the founding chair and alumnus of Indian National Young Academy of Science. She is presently working as an associate professor in the Department of Biological Sciences in IIScr, Kolkata, and she also holds the office of Associate Dean of International Relations Outreach in the institute. She has completed her PhD from Center for Ecological Sciences, Indian Institute of Science, Bangalore, under the guidance of Professor Raghavendra Gadagkar. She has received the INSA Medal for Young Scientist in Animal Science in 2009 and SERB Women Excellence Award in 2013, among other awards and honors. Her research is focused on the areas of animal behavior and ecology. She had worked on the politics of social WASP, ROPALIDIA MARGINATA, and presently she is exploring a new model system, free-ranging dogs in India. She is also a theatre enthusiast and is associated with the theatre group Mukosh. She has accepted our invitations to speak in the webinar in this webinar series, and we welcome her. And regarding the question and answers, we will have the question and answers at the end of the lecture. We request all of you to type in the lecture, type in your questions in the question and answer box, and I will read out the questions. For some reason, we are not able to, put the live video of the speaker today, but you can hear her and see her slides. Anindita?
Yes, thank you, Bithasta. It's a great pleasure for me to be here today. We are sorry somehow the Teams interface and this connection is not working, and my video is not going to be live. But if anybody wants to know what I look like, I think I'm there on the poster. So we live with it, and I'm glad that the slides are visible. I'm sure you can see the slide now.
Bithasta, yeah, I can see. Yeah, we can see.
Thank you. It's a special privilege and pleasure to be virtually, though, be back in IISc to give a talk. So all invitations to deliver lectures are very nice. I like giving talks, but then when your alma mater calls you, it's extra special. So I'm really happy and honored today to be here. What I would like to do is tell you a few stories, because from what Bithasta sent me about this entire series of Paraspar, it appeared that we are more interested in hearing about the backstories behind the research than about the research itself, which you can of course read up in papers and you can hear in umpteen talks. And I really like this idea because when we write journal papers, they are so dry and cut out and written in such a monotonous way that you don't get to tell the story. And especially for us animal behavior researchers, we typically pile up a lot of interesting anecdotes while we go forward. So what I thought is, because I have the chance to do it here today, thanks to Bithasta and IISc, I'm going to begin first by thanking Bithasta for this very interesting invitation. And of course, IISc, it's always a pleasure to go back there. And even though it's virtual, I almost feel like I can, sense the air of IISc by closing my eyes for a minute. And I actually picked this particular photograph of Bithasta from her social media profile among many beautiful pictures, because she happens to be standing in the courtyard of the Center for Contemporary Studies, which has now been renamed, of course.
The slides are not moving.
I've not gone to the next slide?
No, not yet.
Oops. Shall I try sharing once more?
Yeah.
Yeah, just.
Yeah, now we can see. Now we can see.
Now you can see it, right?
Yeah, yeah.
Okay, something. Now it's right, okay? Yeah, so as I was saying, I picked this particular image of Vitasta because she happens to be standing at the Center Studies, a center which I have been involved in from its inception to its renaming and remodeling. A place very close to my heart at IISc, among some other places. Of course, Center for Ecological Sciences, where I did my PhD. So thank you, Vitasta, all over again, and thanks to the communication cell of IISc for the invitation. I'm going to begin with the story of a couple of interesting stories, rather, from my PhD days in IISc. And as Vitasta mentioned, I did my PhD in the Center for Ecological Sciences with working on this very fascinating wasp species, which I'm going to tell you a little bit about in the next couple of slides. This is a wasp called ROPALIDIA marginata, and it's widely available in southern India. So I'm sure people in IISc come across this wasp quite often. And if you haven't ever seen this wasp, then when the lockdown is over, do walk into the West Puri in Professor Gadakkar's lab, and you can catch a glimpse of these wasps. Of course, they don't come with all those fancy colors on them. The colors are put by us to give them individual codes so that we can recognize them individually. And this is a species of paper wasp. So the nest you see that they are sitting on is actually made of paper, which they produce by scraping cellulose from any source available. They can even use tissue paper if you give it to them. And pretty much the way we make paper, so make pulp out of it and lay it out in a thin layer and you get paper. And I have worked and learned a lot from my supervisor, Professor Raghavendra Gadakkar, to whom I think I owe my ability of thinking critically and of asking questions. So this journey began in his lab, my journey as a scientist. And we have been this entire lab. We are a set of people who were in love with this one particular species of wasp. So when I joined the lab, we already knew a lot about ROPALIDIA marginata because Professor Gadakkar had been working on it for about 20 years then. So this is actually a primitive society where a queen does not look like the workers, unlike queens in, say, ants or queens in honeybees, where the queen there's morphological difference between the queen and her workers. There is always only one queen in the colony, and you have to identify her by seeing her lay eggs, no other way. And she is actually a very, very docile animal on the nest. That doesn't mean that everybody on the nest is docile. Workers use aggression among themselves to do what? To self-regulate work on the nest. So workers beat up each other to say, go get food, there's work to do, there's nest maintenance to do, etc., etc. So aggression is a means of self-regulation in the species. And very, very fascinating. When the queen dies, or if you simulate a situation by just plucking the queen off the nest, when the wasps think that the queen is dead or gone, then only one of the workers existing in the colony suddenly steps up her aggression many folds, about 80 folds increase in aggression. And if you don't put the queen back, then it takes about a week on an average for this one individual to start laying eggs, by which time she lowers her aggression and starts laying eggs. And this individual has been named the potential queen until she becomes the queen in the lab. When I joined the lab, from the work of my seniors, all of this was known. This was 2001, exactly about 19 years from now. So August 2001, and there were two burning questions which we were discussing in the lab. One was, how is the future queen decided? Because people had done a large series of experiments trying to see if the potential queen who becomes aggressive after the queen is removed, you then go back and check her state was present. Was she more aggressive than the other workers? Was she the most active among the other workers? Did she have better developed ovaries than the other workers? Was she different in any way? And no matter how much we played around with the system and how brilliant the experiments were, we could not find anything by which we could identify the potential queen in the presence of the queen. So we wanted to know, how is the future queen decided? And very importantly, how do the workers know that the queen is gone? She doesn't look different. She doesn't do much other than laying eggs. She's a docile individual. She's not interacting with anyone. So these are questions which were being actively discussed in the lab at that time. And the senior most student in the lab, Sumana, who now happens to be my colleague at IISc Kolkata, Professor Sumana Annagiri, was in her final phase of her PhD, who had been trying to address some of these questions, and the answers were, we don't know, we don't know, we don't know, and it was very, very frustrating. But Sumana had done some very, very interesting experiments with the wasps. And then there was Sujata. So she was really not working only on Ropalidia marginata. She was working on a related species, Ropalidia cyathiformis, and between the two of them, they were. And then, of course, introduced the junior, the first year, who was wide-eyed and listening to everything that everybody is saying and asking questions. Now, Sumana was asking this question of how do the workers perceive the queen. So she was doing this experiment in which she showed that the workers do not perceive the queen when the queen is across a wire mesh partition from them. So what she had done was she had taken a nest and split it in two. One side had queen and half the workers, the other side had only the remaining half of the workers. We call this the queen right and the queen left sides. And we always found a potential queen on the queen left side of the nest. So there was a mesh in between. Queen and half the workers on one side behaved as if nothing was wrong. The other half of workers had a potential queen beating up all the workers. So what was happening? These were the questions that the reviewers were asking. Why mesh? Why not a solid partition? Why don't you try other materials? Maybe the iron is doing something, it's absorbing something and the smell is not going through. So all kinds of questions to which we didn't have answers. Excuse me. This is the stage at which Sumana was when I joined the lab. And some work had been done and some more experiments needed to be done. And Sumana was trying to finish her thesis. And so it's a typical tradition in the lab that when you need an extra set of working hands who can quickly help you so that you can pace up your work. And of course, in a situation like this, if you have someone who has not started her work yet, but is eager to help, there's no better person to ask help from. So that's how I got inducted into studying WASP politics in my very first year at IISc. We did a set of experiments with Sumana, Sujata. I had a lot of fun learning how to handle WASPs, how to do experiments. So I was juggling courses and assignments and exams and experiments. It was like a hectic 6 months in my second semester, and it was immense lot of fun. So we come back to the question of how is the future queen decided? And I said, okay, fine, we still don't have the answer, but this is something we can explore. But I decided to kind of twist the question a bit, and I said, we don't know the answer to this question, but do the wasps know? I mean, are we sure that there is a pre-designated queen who becomes the potential queen when the queen goes, and then she eventually becomes the queen? I mean, we see the process. You remove the queen, a PQ comes up, and PQ beats up everyone. Everybody accepts her beating and stays on in the colony, and then eventually she reduces her aggression and becomes the queen. Fine. But how do we know that the wasps already have decided that this wasp is going? How do we know it is not something completely random? Whoever starts beating up everyone first, maybe, whoever gets to know that the queen is gone the earliest, maybe gets the chance to be the PQ. We don't know. So this is what I thought of asking. How do the wasps get to know that the queen is gone? What is the mechanism? And do the wasps really have an heir designate, or what I called a cryptic successor in the colony? We don't know who she is, but the wasps know. And of course, the other question of how the queen is perceived by the workers in the colony. And that's how the hunt for what we thought was a queen pheromone, a special chemical signature in the body of the queen, which she somehow releases and the workers perceive, which tell them that the queen is here and everything is hunky-dory. Now, this is not something that I drew out of my hat, of course. People had been discussing about the possibility of a queen pheromone in the lab for quite some time. And we knew that in many highly organized insect societies, like in the ant societies and the honeybee societies and the hornet societies, queens have pheromones. And that is what the workers use to smell or perceive the queen. So why not in our species, where the queen, unlike in many other species, is not very active, does not go around poking and prodding others in the colony, but sits in one place, walks around a bit and lays eggs. You know, poor me, queen. How does she signal that she is present in the colony? And that's how, by the end of my coursework, I started working on my PhD. And the fun began. So every PhD is a learning experience, and I did learn a lot during mine. So when we started thinking about experiments, the obvious place to start were the various comments that reviewers had given to Sumana's work, or questions that Professor Gadadkar had received from members in the audience when he presented this work in different meetings and conferences. Things like, you know, why don't you change the wire mesh with something else? Or maybe, you know, the pheromone is there, maybe it's not volatile enough, so the workers are across the mesh, they're, you know, a few centimeters apart, they don't sense the queen. Maybe they actually sense it by touch. So these are the questions which were there in our head. Is it touch? Is it volatile? Is it semi-volatile? And what do? You design experiments, because the wasps will not come and tell you. They can come and sting you. They cannot come and tell you how they perceive the queen. So we did a series of experiments, one after another. All of them failed, but I learned a lesson, which is not all brilliant ideas lead to publications. And this is very important for every PhD student to remember, especially today. You know, when I was a PhD student in my first and second year, I did not even care whether I'm going to get a paper out of what I'm doing. Frankly speaking, until I went to IISc, I did not even know that, you know, you have to publish papers out of your research. You do research, yeah, you get to know things. Yeah, what do you do with it? No idea. I came from a university system where we didn't have in those days master's projects and BS projects, whatever. You do classes, you write exams, you get marks, and you go on to the next step. So, and impact factor was something I got to hear of only, I think, in my 4th or 5th year in IISc. These days, I'm amazed when, you know, first year students tell me, I want to write a paper, and first year, not PhD, first year BSMS students tell me, I want to write a research paper. And I'm like, excuse me, what are we talking about? Do you even know what research means? So those were the glorious days when I didn't care whether I had my name in a publication or not. I was more concerned about what is the next experiment that we are doing and what fun it is to go collect wasp nests, come and bring them, put them in the vespiary, mark the nests individually, keep following the wasps every day, take attendance. That's what we used to do a census of the nest every alternate day, update the nest maps, see whether the queen is healthy, whether she's laying eggs, feed the wasps, A lot of work to do. Who cares about papers? And this is a very, very important thing. I think we all need to fail, because that adds learning curve. The experience of it has remained with me forever. So we tried out an experiment where we replaced the iron mesh with the nylon mesh, and I think I did about 3 nests, and finally I gave up, because at the end of the day, a PQ emerged on the queenless side. So whether you give them an iron partition or a nylon mesh partition, the workers simply could not perceive the queen if she was on the other nest. Then we said, okay, maybe the queen's pheromone is not volatile, so you have to do something to help. So we actually tried to use a fan to blow air from outside the nest. So there was the nest, there was a fan outside the nest, outside the cage, so that the air would blow from the queen right half to the queenless half of the cage. Now it's easier said than done. You just cannot get a table fan and put it outside the cage of a wasp nest, because the wind speed is going to be just too much. It's going to blow over the nest. The wasps are going to hate it. So what do? And this is where I was doing the actual experiment. I was trying to figure out how to do this by setting up a wind flow without harming the wasps, and yet creating a flow good enough to travel, help a pheromone to travel from this half of the mesh to this side of the mesh to that side of the mesh. And what did we do? We went to KD Market, looked up all kinds of fans, and finally we settled for a medium-sized computer fan, brought it back. I went to the workshop, got it mounted on a stand with an adjustable height so that we could adjust it according to the cage sizes. Set it all up, started my experiment, and realized that if you put this fan up there in the setup that we had made, because it is small, there's a collimated breeze which is literally pushing the wasps off the nest. Even though we felt that the breeze is not enough, it's not strong enough. We were, then we needed a regulator, and I needed to understand. So RG said, probably you need a better understanding of wind flow. So I went to Professor Mukunda, because RG said, oh, go talk to Professor Mukunda. I went to Professor Mukunda, and then he sent me to the wind tunnel lab in aerospace engineering. So at the end of it, the result was we still got a PQ on the queenless side, but the perks. For the first time, I got to go visit the KD Market. I wouldn't have gone there. Only physics students and engineering students go there, not behavior biology students, to get electrical supplies. Putting the fan together, figuring out, what size, what dimension, what speed, how to regulate the speed, blah, 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 all of that. Actually getting to visit the wind tunnel, it was great fun. And learning about the stuff which I would have never read up about on my own. I spent a lot of time, I think 3 or 4 months on this. At the end of the day, the fan became a useful thing for people who were really sweating it out in the Vesperia and there was no air, or it was very suffocating. So you could actually put the fan facing the experimenter and away from the nest and give the experimenter some air. That was it. So it remained with us in the Vesperia until I think the end of my PhD and beyond, but as a fan for humans rather than fan for the fan experiment. Then we had a brilliant idea. I absolutely loved the design of this experiment. Sujata and I did this. We spent a lot of time figuring out what to do because somebody told RG, maybe the way the queen walks, there is some vibration which is specific to the queen, and that is what they are sensing. And of course, we had to try it out. How can you just leave that question unanswered, though we didn't really believe it. But we still had to do something. Now, how do you stop the nest from vibrating when the queen walks? Or from the workers, how do you stop the workers from perceiving this vibration. And just to tell you, this was of course a failed experiment. So at the end, we are going to get the same result. The PQ will emerge on the queenless side, but that's not the point of it. So we did this vibration experiment, and I had immense fun putting this slide together because I actually dug it out from my presentation, which I had prepared for my comprehensive, which it turns out was on 20th of August, 2003. So we are very close to the date, and it was a very interesting coincidence, which I thought was very funny. And this is actually, these are 2 slides which I've taken screenshots of and not edited for the sake of memory. So this was the vibration experiment. You have a whole colony, you observe it for 6 hours on the first day. Next day, instead of cutting the nest, what you do is you make a hole in the mesh. And then you insert the nest through this hole so that half of the nest is on one side, the other half is, and you do the experiment as usual. And the graph here basically shows aggressiveness of the PQ and the queen. On day 1, the PQs are hardly aggressive. On day 2, you have a PQ1 on the queenless side. Day 3, we actually exchange the queen from one side to the other, and on the other side, we get another PQ. And this is exactly what happened in all experiments that we did. Just to tell you about this particular experiment, I said we did the reverse. We did not cut the nest. We cut a hole in the mesh. It's easier said than done. We were using an iron mesh, and early in the morning, Sujata and I would go to the vestibury and start this entire process of cutting the hole, which would fit the size of the nest that we had, and then adjusting the spaces, extra spaces made by using extra wire. So imagine a nest which is irregularly shaped, and then you have to cut it into 2 or divide it into 2, and you pass it through a mesh and you prop it up on sticks and you fix it. And all of this you do after removing the adults and keeping them in small vials. But you don't want the adults to be stressed out. So all of this needs to be done very quickly so that the adults can be put back on the nest very quickly. Now, this is the wire mesh with the hole, and the nest is passing through it. And the nest is in such a way that the mesh on both sides almost touches the nest, but does not completely touch, so that the vibration cannot propagate through the mesh. Important thing is there should not be enough gap for the wasps to go through. So the wasps cannot pass across the mesh, but the mesh does not touch the nest surface. So we thought we had done it perfectly. What could go wrong? And for the first experiment that we did, nothing went wrong, other than the fact, of course, that we got a PQ as usual. Nothing else went wrong, and we were very happy. We went and told RG, you know, the vibration experiment is working. He said, oh, you didn't get a PQ. He said, we got a PQ. Don't worry about that. It's all fine. But the setup is working, and we were very happy about it. But then we had not accounted for the resourcefulness of the wasps and how keen they are on working. So when we did the second experiment, everything went fine up to the first hour after cutting the hole and putting the nest and starting observations. Then I see there is this one particular wasp who is supposed to be on the queenless side. She goes out, gets food, comes back, feeds the larvae on her side of the nest, goes back, gets food, and then she's trying to put her head through the mesh to feed the larvae on the other side. And of course, she can't do it, and she keeps trying. And guess what? After half an hour of this, I see she has happily dug a tunnel for herself because this is a paper nest. She has actually chewed up some parts of the nest so that that part has become low, and there's a nice tunnel through which the workers can move this way and that. And of course, the entire experiment fails because if workers can dig a tunnel and pass through, then you have no way of preventing them. After this, we decided there's no point because, of course, the potential queen was emerging. Vibration is not really something that we should run behind. And given after all of this brilliance that we had put into designing and doing the experiment, the wasps fooled us. And we said, okay, let it be. Another experiment did. Remember, I had almost finished 2 years of my PhD. 1 year in coursework, another year doing field experiments. I did pass my comprehensive and become an SRF, but I still did not have my own data, which would go into a thesis chapter, and I was not panicking. Then we had the T-board meeting. Of course, everybody from IISc knows how important visits to the T-board and coffee board were in those days. Of course, now you don't have the tea board and coffee board, but you have Prakriti and you have other places to hang out in. In those days, we had the tea and the coffee board, and RG, Professor Gadadkar, was not a regular visitor to these places. He was too busy. The coffee typically he drank in his office. He would not really walk to the tea board or coffee board for tea or coffee. But this was one particular day on which we were breaking our head, Sujata, RG, and I. We were breaking our head over what to do next. And he said, why don't we just walk to the tea board? And we actually went and walked to the tea board. And it's actually at the tea board that I still remember. We were having this chocolate pastry cut in half. So RG bought us the pastry and I cut it in half. And Sujata and I were having this pastry, and that is when this entire experiment design popped up. So again, we were discussing, do the wasps know? When I said, we are doing all of this, but do the wasps really know? Is there a cryptic successor? First response from RG was, that's not even a valid question, Anindita. How can you say, how can wasps know? And after a while, we all converged to this idea, well, we can still give it a try, but how? The wasps will not tell us if they know. So you have to come up with a design by which you have to make them tell you the story. And this is where theory of probability came to the rescue. We were cutting, as I said, we were eating a pastry in half, and we had already done these mesh experiments, a whole series of them, where I had now become a champion nest cutter. I had even devised a mechanism by which if a nest was not equal, then I could transplant larvae from one cell to another to make the nest equal. All of that I had learned during my series of failed experiments and my experiments with Sumana. So we said, okay, we realized that when you cut a nest in two, you put the queen on one side and the other side has no queen. Imagine there is a cryptic successor. Then there's a 50% chance that she is with the queen and a 50% chance that she is on the other side. Now, on the queenless side, one individual becomes the PQ. If the cryptic successor went to the queenless side, then the PQ is the cryptic successor. But if she had by chance ended up with the queen, then we actually have an imposter becoming a PQ on the other side. And this was what triggered the experiment's idea. I'm not going to give you the details of the experiment actually, because it's a very, very long process, but this is kind of the design that we had. Half the nest on two sides of the mesh, one side with queen and half the set of workers, the other side with only half the set of workers. And of course, by now we knew we'll get a PQ. But then what do? We give the PQ 2 hours to think that she's the potential queen. Then you take her off the nest and put her on the other side and take the queen from the other side and put her back on the side that the PQ1 was on. What would happen? The workers who will get back their queen will go back to work. Excuse me. But the PQ1 will be with a new set of workers. There are again two possibilities. If she was the cryptic successor, she will continue to be the PQ. If she was an imposter, she will now face the real successor and be expected a fight and a PQ2. And this is where I said we need a third session. Why? Because the PQ2 has to prove that she is not an imposter. So you give her 2 hours and then you take her to the other side and remove the queen back to this side and let PQ2 hold herself with the new set of workers she faces, and we should never have a PQ3. 2.5 years of work, 19 trials that I could remember, maybe some more, I don't remember. Many, literally back-breaking hours of work, because day 1 was easy. I do 5 hours of observation on a normal nest. Day 2, I literally came early in the morning, divided the nest, put everybody back, give them time to settle, and then I had 6 hours of observations with 15 minutes in between. So you start at 8 a.m. and you finish by the time you finish at 6 p.m. and you are sitting on a stool with your nose glued to the cage door, literally watching 2 nests at the same time, where constantly a lot of things are happening. So a lot of hours of observations. Sometimes experiments failed. You try to remove the PQ, she escapes. You try to remove the queen. Some of the workers escape or they don't want to go back to the nest and you cannot continue with the experiment. That's why so many trials were required until we got 8 successful trials of this experiment. And just to sum it up, in half the set of nests, we got PQ1 only, no matter whether you took her from this side to that side. In the other half set of nests, we got a PQ1 and a PQ2, but we never ever got a PQ3. And we actually concluded that we know that the wasps know that they have a cryptic successor. And this we got a major thrill out of when, after some rounds of coaxing, the editor, Biology Letters, actually let us publish this paper with this title. We know that the wasps know, because that was the thrill of it, that we had finally ended up getting to know that the wasps are aware of who their next queen is. After so many years of work put in by so many people and so many failed experiments, we finally had a success story. And this was something that we were really thrilled about. Note the date. This paper was published finally in 2008. And this was September 2008. I submitted my thesis in November 2007. So by the time I got my PhD in April 2008, this work was yet to be published, still under revision. That did not stop me from getting my PhD or doing other work, but this is something that anybody tells me, close your eyes and tell me the best experiment you have ever done, I would say the QPQ exchange experiment, and I never got this paper published during my entire PhD tenure. So to the PhD students out there, hang on tight. If you have a good idea, it's going to click. Don't get frustrated over a publication. So this was finally, the paper came in 2008, as I said, 2 more years after I had finished the last experiment. A lot of data analysis after receiving my PhD and having given birth to a son in between. The other story I want to talk about in brief started somewhere around late 2003 to early 2004. Many of the students before me, especially Sumana and Sujata, had reported that the queen shows a very peculiar behavior on the nest, which we called the rub abdomen behavior. And it was fun because we used to say are, but RG used to say no, but that is abdominal wagging. We have already reported it. It's nothing new. And we always said no, sir, but it's something new. It's something different that we have not recorded earlier, and we should pay some closer look. So it was really funny because when a new student comes to the lab, we go through a process of doing what we call ad libitum observations. So you go and spend your time watching the species, making your own notes in as much detail, and kind of getting to know the system. And then at the end of it, seniors will tell us what you saw after 50 hours of observations, after 60 hours of observations, and so forth. So I remember during my first month, Sujata had told me, whenever you come have time, you can come and sit in the vestibule and do ad libitum observations. What she had forgotten to tell me, and this is something I always now make a note of with my students, we very categorically tell them, whenever you're doing ad lib, even if it is for 5 minutes, please note it down. Sujata had told me in passing because I think she was not sure I would be starting in my first semester, because typically in the lab, the first semester, new students were allowed to be on their own, do courses and not stress out with work. So she had said, we'll be doing ad libitum, and I used to go up to the vestibule and sit in front of the nests and keep seeing what is happening. And then I came and told Sujata one day, there is this peculiar thing. There's one wasp on this nest who keeps walking as if she is, painting the nest with the tip of her abdomen. She's almost rubbing it like a brush. What is this thing? And I still remember the thrill. Sujata jumped up and went running to call Sumana and tell her, hey, see what this new kid has seen. And I was like, what on earth are these crazy people talking about? It was later that I realized that they had not told me anything, because the idea is you start with a blank slate. And they were thrilled that a new observer had noted the rub abdomen behavior, which is something that they were very kind of enthusiastic about. And they went and told RG, see, even this girl has seen rub abdomen, we have to do something about it. So what we had was that the queen shows this behavior most often. We thought so. And our hypothesis was that the queen has some gland from which she's producing some pheromone, which she's actually rubbing on the nest as she's walking. Imagine you are a docile individual. You don't like shaking hands with people. You don't like, talking to people much. You don't like interacting. But if you wear a perfume, people know you're in the room. It's like that. So the docile queen doesn't have to interact with workers. She can just walk on the nest and rub her abdomen on the nest, and her smell will continue to be there on the nest, which the workers can perceive. And we said that the gland must be opening at the tip of the abdomen near the base. So we said we have to search for possible candidates for the gland, and came up with one candidate, which we thought was quite a close match, which is known as the Duffer's gland, has been reported to have various secretory functions in other species of wasps and bees. So we wanted to carry out a detailed study of the rub abdomen behavior. We had to convince RG that something's happening. So again, this was partners in crime, Sujata and me. And we said, we need a bakra. So literally, Sujata and I, one day, we barged into RG's room and said, we need a bakra. And he was like, what's that? So we said, we need an enthusiastic junior who is ready to do an experiment that is a bit tedious, but it needs to be done. And he was like, okay, where do I get one? But of course, before going to him, we had identified the bakra. And we had decided that we want to plan and execute what we call a bioassay, an experiment by which there is a biological signature which tells us something about the system. And the bakra was Shobhik, who was then a summer intern in the lab, a very enthusiastic undergrad. And we had decided we are going to catch hold of him. We only needed the permission. That's why we went to RG. So we had Shobhik, who perfectly suited the plan. So we made him do this experiment. This is my nest map from one of those nests, and his job was to sit and watch who is doing rub abdomen, and mark the trajectory like this on the nest map with a pen, showing where she is walking, and writing down the starting time of the behavior, ending time of the behavior, and recording each bout of it. As I said, it was tedious work, not hard, just time consuming, and sometimes very boring if rub abdomen never happened. 2 months of work, several nests were covered with several days on each nest. And the conclusion is queen performs this behavior about once in 18, and the workers rarely showed this behavior. Why I'm telling you this story is because we never published this piece of work in detail. We just used that 18 minutes in a different paper at that time. But this particular work, which was tedious, backbreaking work, interesting design, and led to a lot of new ideas, never was written up as a paper by itself. But this work acted as the foundation for a lot of future papers from the lab. We did find the queen pheromone at the end in the Duffer's gland, and people in the lab after me continued to work on the Duffer's gland and have done excellent work, especially Aniruddha Mitra, who has worked extensively on the queen pheromone. So the lessons learned, publishing a paper is not really the goal. Just one of the results from the piece of work that you do. It is the experience which counts. And another lesson learned, an enthusiastic intern can do a lot of work in 2 months if there is good planning and some mentoring available. And this lesson I have kept with me all my life. In my lab now, I always have a whole lot of interns. Right now, there are 10 students doing virtual internships in the lab. And many of our papers from the dog lab have a long list of authors, not because those are our MS or PhD students, but more than half of them are often summer interns who have done good work during their 2 months of stay in the lab. Of course, under the mentorship of some very efficient PhD students. So in IISc, I was there for almost 8 years. I went in 2001 and I left IISc in June 2009. And I learned a lot of lessons for life. The person I have learned most from is Professor Gadekar, who taught me the importance of asking questions, importance of mentorship, internships, training others, outreach, and breaking boundaries between disciplines, especially through CCS. And this is very, very important because later in life, as I engage in different activities, I see how important each of these lessons have been, especially how important training others have been. From IISc, I came to IISc Kolkata, and in my first semester here, I started teaching an undergraduate course. And I never found it to be a difficult task. I do all kinds of courses here, and all the teaching that I do, the backbone of it came from my handling of hordes of interns over every summer that I stayed in the lab, and training juniors and other, talking to visitors, telling them about work. There was a point of time when I used to joke, you wake me up in the middle of the night and you ask me a question about the lab and I can give you, a briefing even without waking up completely. And that is what I think every PhD student should be able to do. Every PhD student in the lab should know in detail about every other student's work. I often see there are people who don't talk to each other about their work. They don't discuss their work with their lab mates, with friends across labs, and this is not done. You cannot do your science alone. Your science cannot be clustered. And these are lessons which I have learned in my tenure at CES. We were a very open place. Everybody went to everybody's talks. All comprehensive exams, all thesis colloquia, all thesis defenses, all ESS talks were full, and everybody gave comments, everybody understood everybody's work, and that is how science should be done. So from IISc, I came to IISc Kolkata, and I went through a process of academic weaning before I came here. So after my PhD, I was continuing working on similar questions on still ongoing projects as a postdoc. And I was asking myself, what next? Of course, the easy choice was a postdoctoral experience abroad, but the difficult choice was staying back in India. With this came the choice between career and family, because as I said, I already had a son who was very small, and the choice between what people call success versus my personal happiness. And again, RG came to the rescue and suggested, why don't you try applying to the IISers, because these are new institutes. At that time, there were only IISer Kolkata and IISer Pune, and they are hiring a lot of young people. Great, why not? But then what should I work on as an independent investigator if I apply to IISers? I will need a work proposal. I came up with 3 mini proposals and I put them on RG's table and I asked for comments. So after working on 3 mini proposals and having a long discussion with RG, I finally had my model system. Here I want to stress that I have been lucky in having the absolutely perfect mentor in RG. Even today, something goes wrong, the first person I think of calling is him. I pick up my phone and call him at 10 o'clock in the night. And this is something very important, I think, in the context, especially of Indian researchers. When people heard that I'm working on dogs, often they asked me, how come you're working on dogs? You have worked on wasps. And then the second question would be, what did Professor Gadagkar say? So I'll tell you what Professor Gadagkar said. When I went and spoke to him, he asked me why I want to work on something different. What is the logic behind my choice? And I told him that if I'm going to stay in India, I don't want to work on social insects because of a very simple reason, not because I'm not interested in social insects, but of the different available options in India, I'm more fascinated by wasps than other insects. If I want to work on wasps, I will have to start and follow the trajectory that RG had taken because nobody could have done it better than him. Logically he has developed his work on ROPALIDIA marginata over the years. And then I will be simply copying him onto another system. And I told him, I don't want people to always identify me as Gadagkar student. I want to be me. I don't want them to just say, oh, your student is just a replica of yourself. I look up to him, and because of this, I don't want to replicate his model system onto another species in another place. And I can vouch for this, that most supervisors, not just in India, but everywhere else, would have been very upset. Most supervisors want their students to be replicas of themselves, to propagate the network, to cite each other, and so on and so forth. There's a whole lot of discussion which goes on this topic. But my supervisor told me, you are going to give me true academic fitness because you are going to start a new field of work. I couldn't have asked for anything better than this. And this is what a mentor's role is. A mentor should not be telling people what to do, but a mentor should be the guiding star. And that is what my mentor has been for me. So advice to PhD students, again, find a mentor. You will need the mentor all your life, and you're going to be happy if you have a good mentor. We came to dog watching. I started in 2009 in IISer, and in the beginning there were Srijani and Manubi. My first 2 project assistants turned students, and then there were dogs. The good thing was they're there everywhere. So one of the reasons why I decided to work on dogs were I would not have to worry about which forest to go to, which forest official to run behind, where to get the permits. You step out of the lab and there are dogs. I knew I would be teaching a lot. I did not want to go around doing field work, taking time off teaching, because I knew in IISer it would be difficult. So that way the dogs were an excellent model system. In addition to that, they were very interesting to study because everyone in the world was studying pet dogs, trying to use pet dogs to understand the evolution of dogs, but free-ranging dogs or stray dogs or dogs on streets provided a much better model system for understanding the evolution of dogs. So we studied on the IISc Kolkata campus. The picture here at the bottom is actually a photograph from IISc campus where I actually started my work. Before coming here, I was doing pilots and developing my research proposal based on the pilots in the IISc campus. Past, present members of the Dog Lab who have been the dog watchers and who continue to watch dogs, and of course, now have lost count, at least 50 or more interns and summer students who have been in the lab, external MS project students who have all contributed to the dog watching years over these past 11 years. And here I'm going to just briefly tell you some of those stories. We started noticing oddities. Manavi came and told me one day that in a group there are two mothers and the pups are confused which mother belongs to whom. What do we do? I said, keep watching and keep noting. Mother is not sharing food with her pups. Is there something wrong? Should I stop watching this group? I said, no. What's the age of the pups at which the mother is starting this? Is the mother still nursing or has she stopped nursing? Is this happening in other litters? Go watch. Somebody came and said, several mothers are showing this behavior. Both Srijani and Manavi saw this. And they are also refusing to nurse. And I had an idea. Are we seeing parent-offspring conflict in the wild? If so, we have a fantastic opportunity to test out Trevor's theory. So I said, okay, keep watch, go read this particular paper and come back. Next day, Manavi comes running, brimming with excitement. I think this is what we are seeing. What do we do? And we plan what we call the parent-offspring conflict experiment. Very simple, give food to the mother and her pups, see if she allows the pups to feed or not. And we saw that when the pups are younger, the mother will allow them to feed with her. When the pups grow older, the mother starts showing aggression, snatching away food. And I'm not going to go into the tedious details of the experiment, but what we found is conflict is more, even higher when you give them meat instead of biscuits. And this was very interesting because we found the conflict to be context dependent. I will just fast forward this up. So we got a lot of visibility with this work from across the world. And we said, okay, fine, now people know what we do with the stray dogs. We found that the mothers are caring, but they reduce their care as the pups grow older, and after a point, nursing stops. We also saw that at the 9th week of pup age, there is this curious peak. Manavi was worried, is there something wrong with the data? So we said, no, let's explore, let's find out a little more, don't get worried. And we had to again tease the animals to tell us what they were doing. Again, design experiments. In this case, we designed experiments with, I'm going to just skip this. So we said, record the details of each bout of nursing or suckling that you see. Ask who initiates, who terminates, is there aggression, what is the duration of each bout? And several, 3 years of data went into this when we said that in the later phase of the pup's development, all suckling bouts were initiated by the pups. And in the 9th week, this is when this is starting, then the pups are trying to grab the mother and suckle from her. And this is what is leading to this problem of the peak in this behavior of the mother in the 9th week. Ignore the statistics and the details of the graph. What I want to highlight is it is also around this time that the duration of mother-initiated suckling reduces, and the blue ones, the pup-initiated suckling goes up, and after a point, both parties agree from the 14th week onwards, no more suckling is necessary, and this very nicely fitted, the two graphs fitted together, and we found support for the offspring conflict theory over weaning. And we identified the 8th to 13th week of age as the weaning period in pups, but realized that for this, Manavi had to keep record of each and every bout of nursing or suckling that was happening with each of these details. Only then, beyond this, statistics came to the rescue. And there were females in the lit group who are not mothers but relatives who were also seen to suckle the pups, which was very odd. But then when we looked closer, none of these suckling bouts were initiated by the allo mothers. All of them were terminated by the allo mothers. And we concluded that it's basically the pups which are stealing milk from any available lactating female because they are hungry. So we actually got a major kick out of publishing a paper titled The Great Indian Giant Families of Free-Ranging Dogs. And we reported selfish mothers, really selfish mothers, selfish puppies, and the giant family system where everybody is trying to help each other. So complex dynamics of dog social lives revealed through 5 years of work actually, which went into Manavi's thesis, again, through the designing of experiments, but the most important part was one observation leading to a question, leading to an experiment, giving an answer, opening up more questions. And that is how our research has been progressing through a series of questions and answers, which when strung together gives us a story from the animal's life. There are stories behind stories. For example, when students get grabbed by people in the neighborhood, taken to the municipality or to a local community center, and I get a call asking, do you know these people? They are roaming around in the neighborhood with the video camera and giving food to the dogs. We think they are thieves. And then I have to go to the rescue. So now my students carry a letter from me saying they are bona fide students of IISc Kolkata. They are working on a project on dogs, which requires feeding and taking videos. So please allow them to work in your neighborhood. Not that that stops them from getting into trouble. Then we have the problem of the possessive dog lovers. One of my students gave me a frantic call at one evening over a weekend saying, you know, talk to this guy. And there's somebody shouting over the phone. And finally, I figure out the student was doing an experiment which involved feeding the dogs. And this was like an experiment in which he was offering food, small pellets of food on a kind of cardboard piece, trying to look at food preference of dogs. And this guy thought that he was poisoning dogs. And then he comes out and says, what are you doing? And he says, I'm doing an experiment. And then he says, you know, I feed pedigree only to the dogs in this neighborhood. You're not allowed to feed dogs here. And then he says, I'm going to call the police. And then the call comes to me. The best story I want to end with is from Srijuni. This is in her 2nd year as a student here. She is working somewhere in the neighboring agricultural university campus doing long-term observations on mating behavior. And typically mating happens when it's also raining. So she has an umbrella. She's sitting under the umbrella and recording. And an elderly couple passes by, giving her weird looks. 1 hour later, the elderly couple is returning from wherever they had gone to. And this time, Srijuni hears the lady tell the gentleman, oh, I feel so sorry for this girl's parents. This poor girl, she's so young and she's gone mad. Imagine what the parents must be thinking. So these are the traversities that we have to go through, interesting experiences. Someday I'm going to write a book about it, of the stories that we accumulate while we do fieldwork. I'm going to stop here. Before I stop, I really want to thank again IISc, Vitasta especially, IISER Kolkata, INSA, CSIR, SERB, the work over the last 11 years and for the support that they have given to us, and all members of the Behavior Ecology Lab, which happens to be the group that Sumana and I run together, half of them working on ants, the other half working on dogs, continuing the tradition from CES. Thank you.
Thank you, Anindita, for this intriguing lecture. To be honest, when we conceptualized this lecture series, we wanted exactly, this kind of lectures where the speaker not only tells us about the research she is doing, but also the nitty-gritty of it, the trials and errors, the, not so certain methods, not so certain journey of reaching something certain. So thank you for laying it out so honestly for us. Thank you very much.
My pleasure.
Now we'll take some questions. Questions are now open. You can, the attendees can type in the question in the, question and answer box, and I'll read them out. Also, please type in your name so that we know who it is.
Okay, I can see a question from, I don't know who. Shall I read out the question, Vitasta?
Yeah, sure, but I can't see. I don't know why. I can't see the question. Now I can see.
Okay, I need to publish the question, then you can see it.
Okay.
So the question is, compared to then, is there more pressure on students now to publish a lot? If so, is this preventing them from pursuing some interesting ideas just because they may not be considered impactful? I don't think it's then and now. I mean, I don't think I'm that old. I don't have that much gray hair yet. That I have gone through a lot of transitions. I think, well, if I think back to those days in IISc, in general, I think people have become more competitive, and the pressure to publish, pressure to accumulate impact factors and look at indices has gone up. Also because I don't blame the students for this, because there's this entire notion of hiring, which is completely topsy-turvy in many places across the world, including our country, where people don't look at the quality but the quantity. So of course, students are also in this rat race. They have always been in the rat race, but not by choice all the time. And there is also a lot of pressure from faculty on the students to publish more, because the faculty's promotion, the faculty's next grant or the award depends on it. So point is, I don't know if it was there even when I was a PhD student. I'm sure it was there in some places and some labs, because I knew of friends who were highly stressed out. But I think I was just lucky to be in a lab where my supervisor was always happy when I came and said, I'm in the students council, I'm editing voices, I'm doing this, I'm doing theater, I remember after my colloquium, somebody actually came up to me and said, when did you do all that work? You were always doing extracurricular activities. So that was the notion I gave people, and I had a supportive supervisor. Not everyone has a supportive supervisor. I have had friends who had a lot of trouble because their supervisors were upset because they were getting married or they were having babies. My supervisor was the first person in IISc who got to know that we were expecting a baby. So that makes all the difference, I think. It's not really the attitude of the students, but also the attitude of the supervisors, which matter a lot.
Yeah, I can see two more responses, but those are only two,
Yeah, they're comments.
Yeah, comments.
Excuse me.
Any more questions? Okay, we'll wait for like a few minutes, then we can